Thank you. Thank you for the invitation. It's been a really wonderful semester here. In fact, most of the work I'm going to talk about today is work that actually was done here with some of the participants. So we've heard already in several talks about this issue of getting conflicting signal from different genes when you're doing a phylogenomic analysis. So suppose you're trying to reconstruct the species history of a bunch of organisms. Different genes might support different trees, and we've seen many reasons for that today. One is this incomplete lineage sorting problem, which is this sort of population genetic effect, which I will remind you what it is in a second. Another issue is, as Tandy pointed out, is this issue of gene tree estimation error. Right? So you're reconstructing trees from finite length genes, and you're going to make some mistakes. And it's sort of a fundamental question is to try to understand what's sort of the interaction between these two things. I think Tandy really sort of set this up great for me. Here I'm going to give you this sort of a theoretical perspective. There's been very little or no really work on this question of incomplete lineage sorting and gene tree estimation error. And I'll tell you some sort of preliminary results about that here. Okay, so I'll explain the quantity versus quality a bit in a second. Let me just start by setting things up and reminding you what this incomplete lineage sorting thing is, and then I'll explain the kinds of questions that I'd like to be able to answer. Okay, so we've seen this picture many times today. Let me just go through it one last time, make sure everybody knows what I'm talking about. So what is this incomplete lineage sorting problem? This is caused by the failure of two lineages to coalesce in a population. So suppose that you're getting one gene from an individual in this species. You're getting a copy of the gene from an individual in that species. If you trace back their lineages, right, so from ancestor to ancestor, the sort of implicit assumption in standard phylogenetic analysis is that as soon as you get to this joint population here, these two lineages immediately coalesce. And what happens afterwards, the mutations up there are sort of shared by the two lineages there. But in reality, if you have a large population there, it might take a while for these two lineages to actually meet, right? I'm following their ancestors until they actually choose the same parent. And so in particular, if you're estimating the coalescence time between these two individuals, well, this coalescence time is going to overestimate the actual divergence time, which is this branching of the population itself. Now, things get even more interesting up here if you have a case where maybe the population is so large that these two lineages actually never even met in that population, and instead entered the next population and were joined by lineages coming from another lineage. And what happens in that case is that in this case, right, C might end up coalescing first with a more distantly related species, which is in this case D. Right? So, and the reason this is interesting because now if you look at not only just the coalescence times, but the actual gene tree topology, you'll see that C is now more closely related to D than it is to the ancestor of A and B, even though in the species history, this sequence of divergence time, the divergence of populations, C was actually supposed to be more closely related to A and B. And so in particular, if you're using this gene tree to try to reconstruct the species history, you're going to get a mistake there. So this is incomplete lineage sorting. Now, what we're trying to do here is to reconstruct this bold tree. Okay, suppose that we're interested in reconstructing the species history, which is this sequence of speciation events. We don't actually get to see directly what the species tree is. Instead, right, we're going to look at gene trees. Right? So for each gene, I'm going to reconstruct from sequence data a particular topology and branch lengths, and I'm going to do this for different genes, and different genes might give me different things. And now with this collection of gene trees, this sort of cloud of gene trees, my goal is to try to recover the population, the species history. Okay, so the original species tree. Now, in order to do this in a sort of principal statistical way, now I have to model what is the distribution of these different gene trees to be able to take that into account. And as was said before, the natural model here that's used in this literature is called a multi-species coalescent. And a multi-species coalescent is very simple, right? In each population, you're doing a coalescent, and you join them together in this tree. Right? So in every population, you have a certain number of lineages that enter it, and then at a certain rate, every pair coalesce according to the coalescing process. And then by the end, there's a certain number of lineages that are still surviving. They're going to enter the next population. They might be joined by lineages coming from a different population. And then you start the process over. You have a coalescence of all of these lineages. And you repeat this and so on. So that gives you a distribution for these genes. And so once you sort of specify the distribution, you can look at your gene trees and try to infer which species tree is sort of likely to have generated this particular collection of gene trees. Now, it was noticed, immediately when people started trying to reconstruct these species trees in this context, that that there's some interesting issue, which is that not only is it possible for certain genes to conflict with the actual species history, it's actually possible that a majority of your genes actually tell you something that's different from what the species history is. So this is a so-called anomaly zone. Suppose that you're reconstructing a tree where all of the branches at the bottom are very, very short. What is going to happen? Well, what is the effect of these short branches? Short branches here means that the probability that the lineages that enter here coalesce inside here is very small. And what's going to happen is if I follow one gene from each one of these species, it's very likely that none of them is going to coalesce at the bottom. Instead, they're all going to enter this top population together. Now, once you enter the top population together, what happens up there is just a standard coalescent process. And the coalescent process really doesn't care about what happened at the bottom of the species tree. Right? It just spits out a coalescent tree. And it turns out that the coalescent, the standard coalescent, is more likely to output, say, a balanced tree than a more unbalanced tree, which is what I'm trying to represent here. And so in particular, if you started with a species tree that was unbalanced, well, you're out of luck. The coalescent is going to tend to spit out the more balanced trees, and you're going to get, in fact, you can show that in cases like this, the most frequent topology of your genes is actually going to be different from the species topology. Does that make sense? Okay, so you have to be careful when you're trying to reconstruct to take that into account. Okay, so we're going to say that a method for reconstructing a species tree is consistent is if as the number of genes goes to infinity, the method, which starts from the gene trees and outputs the species trees, that method is guaranteed to recover the correct tree with high probability as the number of genes goes to. And so what we just showed in the previous slide is that this is a result of Degnan and Rosenberg that was mentioned before today, is that if your method is just to look at the most common gene tree topology, you're not guaranteed that this is going to be consistent. Actually, it could be positively misleading. It could give you something that's guaranteed to be the wrong thing as the number of genes goes to infinity. There's an important exception here, which I'm going to use, which is the tree taxon case. If I have only 3 species, as I mentioned, then it turns out that the most common topology is the correct one, is the one that corresponds to the species tree. But there's also lots of other methods for sort of getting around this problem of the fact that the most common gene tree topology is sort of not the same as the species tree. Let me describe briefly a couple of them because I'm going to use them. So for this sort of more theoretical analysis, I'm going to restrict myself, just for simplicity, to the 3 taxon case. Okay, so I have 3 species. Think of it as human, chimp, and gorilla. And I'm trying to figure out which 2 of them are the most closely related. Okay, so is it human and chimp or human and gorilla and so on? And I'm going to assume that I have a molecular clog. These population sizes are all the same. So this is a very sort of nice case. And basically, what's going to happen is that the shorter this branch up here is, okay, the smaller f is going to be the length of this branch. Or you can think of it as the probability that 2 lineages coalesce here. The smaller f is, the more incomplete lineages sorting is going to be, the harder it's going to be to detect which pair is closest. Okay? So here's one way to do this reconstruction. If I give you a bunch of topologies, you can just look at which one is the most frequent one. Okay, so in general, that doesn't work. But if you have only 3 species, that actually works out. And we'll actually show the formula for it. Okay, so this is a consistent method. There are other methods that, instead of looking at topology, will actually take into account branch lengths, in particular the sort of estimated coalescence time. And just to sort of get an intuition of how this works, what I'm showing you here is that, okay, so for these are, maybe this is not the best picture, these are not different individuals, these are just different genes. Okay, so what I'm trying to say is that there's a distribution of coalescence times, right, that depends on this model. And how do you sort of extract information that allows you to distinguish between AB being closest versus, say, BC being closest? Well, there's a natural thing to do here, which is just to, for this pair, for each pair, you just take the minimum of the coalescent times that you're going to get, right? So for each gene, you get a different coalescent time, you get this distribution. The minimum is going to tend to converge towards the actual divergence time under the coalescent. And therefore, over here, it's going to be smaller than it is over there because there the coalescent, the 2 closest one was actually BC instead of AB. Okay, so one way to reconstruct a tree is just to, for every pair of species, you look at the smallest coalescent times that you see across all your genes, and you compare that. And the smallest one will give you which pair is closest. Now, this is guaranteed to be consistent when the number of genes goes to infinity. Another thing you can do is just to average the coalescent times that you see across the genes. Right? So for these 2 species, I have a bunch of coalescent times. I can just average all of them. Now, this is not going to give me a good estimate of the divergence time, but it will give me an estimate of something that is going to be smaller here than it is over there. And if I just, again, compare the average coalescent times, then the smallest one will correspond to the closest pair. Okay, so that's another way, another consistent way of getting the tree out of it. Okay, so these are, in essence, the sort of 3 classes of methods for the 3 taxon case. They reduce other methods that are sort of different for more taxa, reduce to those. So the first one is the one I'm going to call R star, probably the most frequent topology. The second one I'm going to call glass, which is the minimum coalescent time across genes. And the third one is going to be stake, which is the average coalescent time across genes. Okay? It turns out that I will use ML for maximum likelihood. If you're given the coalescent times for all your genes, then the maximum likelihood estimate under the multi-species coalescent turns out to be the same thing as taking the minimum. Okay, so you can prove that in this particular context. Okay, but I'm going to call this glass. Okay, so these are all consistent methods. If you really want to be able to sort of compare them with each other, maybe what you want to do is to look at their sort of convergence rate. Right? So as the number of genes goes to infinity, you're sort of guaranteed to converge to the true topology, but how many genes do you need to get a high probability reconstruction? Well, you can compare that between these different methods. Okay, so this is the notation I'm going to use in the next slide. Right, so for the convergence rate, right, so if I think of the probability that a particular method fails to detect that A and B were the closest ones in this case, it's going to decrease exponentially. So this is the probability of failure. Is going to decrease exponentially as the number of genes goes to infinity. You can show that. And it's going to decrease at a certain rate R, which is going to depend on the method, and it's going to depend also on the strength of the incomplete lineage sorting, how short this branch is. And by comparing these R's, you can figure out which methods are sort of better than others. Okay, so the bigger the R, the better. Okay, so this is what you get if you compute explicitly these convergence rates for different things. So this is a result from last year. I'm not going to go in details over this, but what you can see is that you have sort of 2 different behaviors. There's this, remember, this is R, the rate, the exponential decay rate of failure, versus the length of this branch, or the shorter it is, the harder the problem is. And the higher this value is, the better the method is. Okay, so you see 2 things here. This ML, or glass method, the one that takes the minimum, has this sort of linear dependence on the rate on F. Whereas the other 2, the one that takes the most frequent topology and the one that takes the average across coalescent times, has a more quadratic dependence on F. Okay, so another way to think about this is, how many genes do I need to get a 95% reconstruction probability? Well, this glass method, the one that takes the minimum, is going to require of the order of 1 over f, if this is this small thing, 1 over f genes in order to be able to do this correctly, whereas these guys are going to need 1 over f squared, which is much bigger. Now, why is it 1 over f over here? Because remember, f is the probability that you have coalescence in that small branch. And if you're taking the minimum, as soon as one gene coalesces inside that small branch, you're going to be able to detect that the minimum here was different than the minimum of another pair. And so in order, since the probability is f, it takes up the order of 1 over f to actually see one of them. And that's why you get this convergence rate. Whereas the other one is more of an average, and you need to kill some variance that somehow depends on the square of this probability. Okay, so you can look in more details at what happens in different regimes and compare these different methods more specifically. But what I really want to focus on is, what I wrote here is sort of the noiseless decay rate of failure. Why do I say noiseless? Because it doesn't take into account the fact that in reality, I don't actually get the exact coalescent times for each one of these genes. I really get some sequence data from which I estimate those. And there's going to be some error. There's going to be some noise in my reconstruction of the coalescent times and the topologies that are going to affect potentially considerably this picture. And the question is, well, what really happens there? Okay, so I want to go back and do a more refined analysis that actually takes into account errors in estimating different things for different genes. Okay, so what I have now is I have 3 species, A, B, C. For each one of them, I have a genome. So this is a sequence, which is made of a bunch of different genes. Each box here is a different gene. There's G of them. Okay, so there's going to be 2 parameters now, the number of genes that I have. And I'm going to assume, just for this simple analysis, that every gene has exactly the same length. So that's not true, but it's a good place to start. L here is how long my sequence is. And of course, what happens is that the shorter a particular gene is, somehow the less signal I'm getting from that gene, the fewer mutations I can see in a sense, and the more uncertainty there will be in what I can reconstruct from that particular gene. Okay, so what's going to happen is now, for each one of these genes, from this sequence data, I'm going to reconstruct, say, a tree and some coalescent times, branch length estimates, and from those trees, I'm going to get this species tree. Now, what I just showed you in the previous slide is sort of an analysis of the step from here to there. If you were given the true gene trees, how well would you do in terms of recovering the species tree and how much data you would need and so on. But I really want to also understand what happens in this step and how the two of them interact with each other. And in fact, it's not even clear that I want to do this. Right? It could be that I just want to go directly from the sequence data to the species tree. And there's a question of, well, actually, is it better to do this or not? Can you prove anything? Can you prove anything about it? Okay, so now I want to go back and analyze these methods again, or maybe devise some new methods that will take into account, okay, this entire thing. I said, how am I going to model this? I'm going to model this in a sort of 2-stage process. So this is the obvious thing to do. I first have my species tree that, according to the multi-species coalescent, generates a bunch of gene trees. Okay, so for each gene, this guy is distributed according to the multi-species coalescent on this. There's some actual branch length here that I'm not showing. And then on each one of these gene trees, I'm going to generate sequence data using, say, the Juke scanner model. Okay, so if you don't know the Juke scanner model, it's just a model where you have a certain sequence at the root of this gene tree, and along each branch, at a certain rate, point mutations are sort of accumulating at random, and it generates a sequence data for your leaves, which are where the data is. And so now, from this sequence data, I want to recover the species tree. And I want to understand the same kind of questions, that is, well, is it consistent? Now, in that sense, this is not the same question anymore. I'm looking at different data and looking at convergence rate. How much data do I need in order to get a good reconstruction of my species tree given my sequence data? And now it's a more complicated problem because now it's 2-dimensional. Right? How much data do I need is really, well, there's the number of genes and there's the length of each gene. And you might be interested in understanding really sort of the trade-off between them. Right? The longer your genes are, maybe you need fewer of them, and vice versa. In general, is it better to have tons of short genes or a smaller number of longer genes? What's sort of the trade-off between this? What are methods that work well in sort of different parts of this picture? So this is the goal. And I borrowing sort of Tandy's terminology, I think of this as a sort of quantity versus quality question. And if you think of L as the quality of your signal, right, for each gene, the shorter it is, the less signal you're getting, so now the lower the quality of your data in a sense is. And you might, and you can think of this picture as sort of quality versus quantity of your data. Does that make sense so far? Okay, so let me tell you what we figured out about this. So let me start with the sort of oldest method for dealing with incomplete lineage sorting, which is to just ignore it. Okay, so the first method is to apply maximum likelihood to this concatenated data. What do I mean by this? Right, we've already discussed this, but just to remind you, I have a bunch of genes. Okay, so this is the genome of one species. What I can do is just pretend like there's no differences between them, and I just concatenate them into one big supergene. And I do this for all of my species, so I get, say, 3 supergenes, one for each human, chimp, gorilla. And then I run a maximum likelihood analysis on these supergenes. Now, what do I mean by maximum likelihood? I really mean the sort of misspecified maximum likelihood, where I just assume that the supergene was generated by a single gene tree. So I completely ignore the multi-species coalescent issue. I have just one gene tree generating these supergenes. And I want to find the best fit, the tree that's going to best fit the data according to this criteria. So the maximum likelihood.
What are you assuming about recombination?
I'm assuming, so that's a good question, right? So in the actual model, the way that the data is generated, I'm assuming that these are completely independent. So these are completely unlinked. I completely ignore the recombination inside of it, and I just assume that they completely recombine between them. Okay, so under that distribution, what can you say about this particular method? Okay, so since it's sort of a misspecified maximum likelihood, it's natural to conjecture that it's not going to work, at least in some cases. There's been some empirical work showing that indeed it can be inconsistent, although Tandy showed that sort of the jury is still out on, in practice, whether or not you want to do something like this or do maybe some partial concatenation and so on. What we can show is a recent result with Mike Steele, is just to confirm the sort of the intuition, which is that not only is this approach in general not consistent, it can be positively misleading. In other words, for any sequence length, you can find a species tree such that if you apply maximum likelihood to this concatenated data, where the data was generated under the multi-species coalescent and the Juke scanner model, but I'm ignoring that when I'm reconstructing the tree with maximum likelihood. Okay? Under that method, then you can find a species tree such that this method will converge to the wrong tree with probability 1 as G goes to infinity. G is the number of genes. Okay, so I fix a sequence length as the number of genes goes to infinity. I can show that this is going to be inconsistent. And the reason that it is I can go back to the sort of this anomaly zone where you can have a particular gene tree that's more likely than the actual species tree. In this case, we're actually going to need to look at 6 species, because this is the first time that you get 2 different unrooted topologies. This is how it comes in. But it's the same idea. If you make all of these branches very short, with high probability, you can make it such that these lineages all sort of meet for the first time in that top population. That top population is just a coalescent, and the coalescent will tend to favor certain topologies over others, and it really doesn't care about what you started with. Right? And so in this case, maybe the balance tree is going to be more likely than this one. Now you have to somehow connect this to what maximum likelihood is doing, which is not taking the most likely thing or anything. It just puts all of this data together, pretends like it's coming from a one tree, and tries to reconstruct a tree. What's going to happen there? How can you analyze this? And there's sort of a natural way to do this here, which is to assume, right? Here I'm trying to find a counterexample, so I can assume anything. I'm going to take the mutation rate to be as low, very low. And what happens when the mutation rate is very low is that maximum likelihood basically behaves like something called parsimony, what is parsimony is, it chooses a tree that minimizes the number of mutations that you have to put along the tree in order to explain the data. Okay, so it's a sort of simpler, more combinatorial quantity. And once you've reduced this to parsimony, you can actually explicitly compute the expected parsimony score of the coalescent under the coalescent of different topologies, and you can show that the balanced one will have a lower parsimony score than the unbalanced one, and therefore maximum likelihood will converge to the wrong thing in the end. Okay, so this particular approach may work well sometimes in practice, but in theory, it doesn't work. So it's not a surprising way. Now, as Tandy pointed out, yes.
It seems like this is a really simple proof, right?
Yes.
I mean, is it complicated or is it this simple?
Well, there's technical steps between these.
Yeah, but this is really great.
Yes, it's a simple.
That's
They
great.
made it look really hard.
That's great.
It's just a few pages. Okay. Now, as Tandy said in her talk, not all concatenation is evil. And let me give you a different take on this. There's a natural variant of the stake method that you can think of as a concatenated method. The stake method, remember, is you take an average of your coalescent times. Now, I don't have the coalescent times. What I have is a bunch of genes. How do I estimate the coalescent time of a particular gene? Well, I'm going to measure the distance between them. I'm going to say, compute the Hamming distance between the two sequences. And that's related by a transformation to an estimate of the coalescent time. Okay? Now, in this particular setting, let's assume that I'm not doing this transformation. I can just work directly with the Hamming distance. For each gene, I compute the Hamming distance between A and B. And I take the average of all of those things across all the genes. And that gives me an estimate of something, which I can show is lower for the closest pair. Now, in my particular setting where I assume that all of the genes have the same length and so on, you can actually think of this as the Hamming distance of the supergene. Instead of doing this one gene at a time, I can just count for each pair of supergene how many differences there are. Here I'm imagining sort of normalizing this by the length of the sequence. And that gives me an estimate of something that is shorter for the shortest pair. And so I can think of this, I'm going to call this stake prime. Stake prime is really a concatenated method where it just takes the Hamming distance of every pair of supergenes and just take the shortest one to be the closest pair. And that turns out to be a consistent method. You can think of this as a concatenation method. It's consistent. And in fact, you can characterize, I have to remember what I really want to do is to understand not only whether something is consistent or not, but what's sort of the trade-off between how many genes I need and how much signal I'm getting from each one of these genes. Right? And so you can characterize this pretty well. Just to get some intuition, first let me just show you a numerical computation of the success probability of stake prime. Okay, so this, what am I showing you here? This is a plot of, for various values of G, remember G is the number of genes, L is the length of each one of them. Right? So for each pair of L and G, I'm showing you the success probability of stake prime of identifying the closest pair. Okay, so red means 1, blue means half, or 1/3, depending on how you parameterize this. But let's just say high probability, low probability. And so what you can see is that this threshold where for any particular length of your genes, as you get more and more genes, well, the probability of succeeding increases, and eventually there's this sort of sharp trade-off where you end up quickly having a probability of 1 of success. And in fact, we already know what the answer is as L goes to infinity because this is the noiseless case. When L goes to infinity, you get the actual coalescent time. And there we showed that stake, which is very closely related to stake prime, needs 1 over f squared. Where is 1 over f squared here? I took f to be 2.5%, because the probability of a coalescent inside that small branch is 2.5%. And if you plot this on this thing, you get this f to the -2, which is a very good fit with where this step is happening. Right? And what you can see now is that as the sequence length changes, this particular method is not very sensitive to how long your genes are. Right? So it gets a little bit worse when the genes are shorter, but not by a whole lot. Okay? And so you can prove this rigorously. You can prove that for any sequence length, as long as you have of the order of f to the minus 2 genes, you're going to succeed. Stake prime is going to succeed with high probability to identify the shortest pair. And the reason this is the case is just because if I write the variance with a sort of conditional variance formula where I condition on the actual true gene tree, there's a sort of a term coming from the variance of the coalescent itself. And that thing really doesn't care about how long your sequences are. And in the end, and no matter what your sequence length is, you're always going to sort of get the same answer, which basically depends on how many genes you have. Okay, so this is what you get for stake prime. And probably you get the same thing for stake. I just didn't prove this rigorously. So this is joint work with Gautam Dasarati and Rob Novak at the University of Wisconsin. Okay, so this is one method. Now, what about other methods? There was this other method which was glass. Right? This is the one where instead of taking an average, you take a minimum of the coalescent times. I can do the same thing as I just did. Instead of looking at the coalescent times, I have to estimate them. I'm going to take the Hamming distance between every pair of genes. So now I'm not doing concatenation anymore at all. I have these different genes. For each one of them, I take the Hamming distance for every pair, and I take the minimum of that across the genes. And the smallest value I declare to be the closest pair. How well does that do? Well, that doesn't do very well, and there's been a lot of empirical work showing that this is a terrible idea. And it's not very surprising. Minimum, taking a minimum is not really a very robust thing to do, and it just behaves very badly once there's noise. Okay, so for example, the Giorgio and Degnan in Systematic Biology 2014 showed that for a fixed L, so for fixed sequence length, as you get more and more and more genes, you're guaranteed that this Hamming distance, again, I normalize it by divided by the number of sites. The normalized Hamming distance is just going to converge to 0 no matter what, right? Because what happens is that as you get more and more and more genes, you have a higher and higher probability of seeing one gene where there was just no mutations there. And eventually this thing is just going to converge to 0. So that's a terrible thing to do. And so you're just going to get 0 for every pair, and so you don't get something that's consistent. Now, it's still informative to look at what is the probability of success for different L and G. And you get a different picture here. So this is the same thing, right, with the same probability of coalescence. Red means high probability of success. Blue means low probability of success. This is for this glass method, so taking the minimum of the Hamming distances. And what you can see now is a very different behavior where somehow in order for this to succeed, you need to have a sequence length that's long enough. If your sequence is too short, no matter how many genes you take, this is just not going to work. But as the sequence gets longer and longer, eventually you get to a point where you can actually succeed, at least with some probability, for a finite number of genes. And if I plot the f, how it corresponds to the f, well, first of all, I know what the answer is in this limit, right? So for glass, we said it takes of the order of 1 over f to see at least one coalescence in that short branch, and in order to be able to detect it with the true coalescent times. So this is the limit over there, and you can see that it goes there. And in general, what you need is of the order of f to the minus 2 sequence length in order to succeed with some reasonable probability. And so we can prove that this is, first of all, this is how it compares to the stake method, right? So you can see it's a very different picture. I'll come back to the difference between them in a second. But this is what you can prove, right? And it just follows from classical sequence length requirement result of Candy and others that say that in order for every gene to be sufficiently well approximated that you can see when something has been coalescing in that short branch, well, you need of the order of f to the minus 2 genes, which is what this thing predicted. Okay. Let me move on to what the real question is. So these are these two methods where you can say something. I plotted the difference between their success probability, rescale between minus 1 and 1. So here glass succeeds well, but stake doesn't, and there it's the opposite. And what you can see is that when you have a large number of short genes, this stake method does very well. When you have a smaller number of long genes, this glass method works very well. When both are long, and there's a lot of them, both of them work. And then there's this whole region here where we don't know what's going on. I guess I'm going to call this the mystery zone. What happens over here? Is it that there are no methods who can do well there, or is there a different method that actually succeeded in that region? Okay, so where can we go looking for a better method if there exists one? Notice that there's sort of a natural way to sort of interpolate between these two methods. Right? Here I'm trying to interpolate between these two extremes in my phase diagram. There's a natural way to interpolate between the two methods is to, instead of looking at the minimum or the average, is to look at some quantile. Right? You can think of the average as being sort of the median, the half quantile. The minimum is the sort of the zeroth quantile. And in general, you can look somewhere in between. And it makes sense to do this because if you think about glass, it's sort of minimum is very sensitive to outliers. It makes sense to sort of just reject, say, 10% of the lowest data and then just take the next value and compare those things in order to see which one are closest. How well does that do? Well, there's one issue there, which is which quantile should I choose? Okay, and so that's the question. Let me go fast through this. Which quantile do you want to choose? So what I'm plotting here is that I have two different distances. I'm measuring the Hamming distance for them. And what I'm showing you is the distribution of the normalized Hamming distances. Right? So there's the green and the blue one that are slightly different. If there was no noise at all, I would just get this step function. But because of the sequence noise, I get this sort of smeared distribution. And the shorter my sequence is, the more spread out this thing is going to be. Whoops, wrong direction. What I want to do is to figure out, well, what quantile should I use? And somehow that should depend on how spread out this distribution is. And so if I write, this is the same thing, I just draw you the cumulative distribution function. And if I look at the difference between the cumulative distribution function, that's going to tell me somehow which quantile is going to work best. Well, it's somewhere over here, which corresponds roughly to the divergence time in terms of Hamming distance, expected Hamming distance. You have to be careful, though. Once you look at this, you have to also think about the standard deviation of doing this. And so here I'm taking the standard deviation of looking at a particular quantile, divided by the difference of the CDF, and you can sort of see that at this minimum, I get sort of the best possible value, which correspond roughly, in terms of the CDF, to a power of L to the minus 1/2. It's actually minus 0.4 here on this particular simulation of my CDF. Okay, so this is the algorithm. I'm going to compare the 1 over square root k quantiles of my distributions. Okay, so I reject the first 1 over square root k, 1 over square root L values, and then I look at this particular, and I compare those. Gautam calls this the quest method. And what you can show is that, well, first of all, it's consistent, but let's look at the actual distribution success probability. Okay, so this is what you get in this case. You get a very different picture where now you have this sort of linear interpolation between these two regimes with a slope of minus 1/2. And if you compare this to these two guys, right, you can see that it sort of interpolates between the two. Steak might do a little bit better here, glass might be a little bit better there, but in between, the sort of quest method where you choose the right quantile is the one that does the best. And so you can prove this rigorously. Let me not explain how the proof works. This one is somewhat less trivial, but not very difficult. What you get is that the amount of data that you need, the number of genes, has to scale like this f to the minus 2, but divided by square root L. Okay, so the more data you have, the longer the sequences are, the fewer genes you need. Let me skip the proof. Now then the last question is, can we do better than this? Okay, so what do we have? We have these three methods. If I superpose them, this is the best of the three for each possible value of G and L. Can I push this even further in this regime where I have shorter sequences and fewer genes? And it turns out that the answer is no. You can set this up you can get a bound as a sort of, you could set this up as a hypothesis testing problem where you're testing between these two hypotheses. You're looking at the distribution of these two things, and you use a likelihood ratio test to distinguish between the distribution of this Hamming distance and the distribution of this Hamming distance. And what I'm going to show you here is an upper bound of the success probability of this particular likelihood ratio test, which is sort of the best thing that you could hope to do here. And you can see that it sort of fits exactly or pretty closely what the best of these three methods are. So in the end, this f to the minus 2 over square root L is somehow the optimal trade-off between these two things. And you can prove this rigorously by computing the Hellinger distance between these distributions. This particular part is much more technically challenging, but in the end you can do this, and you show that this thing is really the optimal that you can possibly hope to do in terms of the trade-off between the number of genes and the sequence length. Okay, so just to summarize what I said, this is the phase diagram. If you have fewer than 1 over f genes, there's really nothing you can do. And then above this slope minus 1/2, which comes from taking the log of this, you get this minus 1/2 over here. These various methods work in these different regimes, and that's really the best that you can do. And then if you're below that, you're just dead. Just to conclude, some interesting consequences of this. Right? Suppose that I go back to the question of how much data do I need? If you measure the amount of data that you need in terms of the total sequencing effort, okay, so the number of genes times the length of the gene, G times L, how does that scale with the sequence length? Well, if you do the computation, right, in that previous picture, I can say for every L, I can find the optimal G. If I compute GL for this particular curve, I get that it grows as a square root of L. In other words, so remember that L measures the quality of my data. The higher the quality is somehow overall, the more total amount of data I'm going to need. Or in other words, you're much better off having tons of short genes than fewer longer genes, as long as what you care about is the sort of total sequencing effort that you're doing in order to get the best possible reconstruction. Another consequence of this is that if you're just reducing things to doing topologies from different genes and combining them together, in general, we can show that you need 1 over f squared, whereas if you start directly from the sequence data and go to the species tree, then you get to remove, divide by square root L, which can be much better in general. Okay, so there's a lot more to do here. And one thing that I've done is to assume that all the genes are the same length. Obviously, that's not going to be true. Different genes are going to give you different signals. How do you sort of combine this together? And what can you prove about this? Different genes might have different rates also, so this is sort of related. I've really done the whole analysis based on sort of distances formally. Well, the method, right, based on distances, but you might want to try to prove something about the full distribution of the data. Look at more than 3 species and so on. I haven't had time to talk about the work I've been doing with Tandy, which is to a sort of different take on this, is to model, instead of modeling the sequence data, modeling the estimation errors that you do in the gene trees and seeing how that affects various methods, but that's for another talk. Collaborators, thank you.
Okay, so we have some questions here.
Yes.
How does the mutation rate enter into this? I mean,
Right,
I noticed
so-
there's a 1 over the square root of L. That's okay.
Yes, that's when I say that I take the sequence length and how the mutation rate should come into this. I haven't computed exactly how.
So you can just sort of scale it out. Right?
Yes.
I mean, so
Yes.
it's long enough so you get sort of one mutation for that length over the time you expect the coalescence to be.
Yes. I looked this up. Yes.
Similar question. I was going to say, if you do gene trees with a phylogenetic method, your lengths are going to be in units of substitutions per site. And we know that genes evolve at different rates, right? So
Yes.
if you're picking the shortest branch length, it just means that gene has evolved more slowly presumably because there's stronger selection of
Yes.
that gene.
All right, so that was one of the future work question. How do you take into account the fact that different genes are evolving at potentially a different speed? And so you would have to do something different or try to take into account that. It's not obvious to me how it fits into this picture. That's a good one. Yes.
Yeah, I was actually going to ask that question also, and also, so if a person was going to do this on actual genomes, then your genes don't necessarily have to be genes, and actually one you might prefer to stay away from actual genes because they're under selection. But then, so then there's the question of finding syntenic regions.
Mhm.
So So there's sort of a trade-off, like if you want a really long region, then maybe somewhere in there's a rearrangement and it's not long enough, so then,
Well, this is saying that you want lots of short things anyway, so maybe that goes in the same direction as what you're saying.
right, like what are the criteria that someone might choose if they wanted to do a bunch of so-called genes, how would they choose their supposed genes?
Yes, these are all good questions.
Okay, so if we don't have any more questions, let's thank Sebastien and everyone who spoke today.